She climbs the stalk of a leaf, locks her jaw onto the vein, and freezes. She isn't dead. She isn't in pain. She is simply a prisoner inside her own body, watching as a fungus hijacks her muscles without ever touching her brain. For decades, we looked at ant colonies and assumed we understood them. Tiny, predictable machines, simple insects, following simple rules. We were completely wrong. The ant brain is not a simple machine. It is a chemical battleground, a mind that can be hacked, dismantled, and entirely rewritten. Sometimes, without the ant ever knowing it has happened. Because what is actually occurring inside their heads is far darker than anything we imagined. They lose their identity. They march in perfect circles until they collapse and die. And some, right now, beneath your feet, are being piloted from the inside by something that has no brain of its own. I've spent months inside the darkest corners of entomological neuroscience to bring you 10 levels of biological horror, starting with level 1, a species that has somehow achieved actual neurological immortality right up until the exact second it dies. Level 1, the immortal who falls without warning. She does not slow down. She does not forget. She does not tire. And then, without warning, she simply stops. In virtually every corner of the animal kingdom, aging is a gradual surrender, a slow dimming, reflexes blunt, memory frays. The body betrays itself in stages, not in fidel dentata. The minor workers of this small, globally distributed ant species do something that has confounded biologists for decades. They simply do not age, not in any measurable way. Their mushroom bodies, the paired learning and memory centers of the insect brain, show no loss of synaptic density across their entire lifespan. Their brain chemistry, far from declining, actually improves. Levels of dopamine and serotonin, neuromodulators critical for motivation, sensory processing, and behavioral flexibility, rise steadily as the worker matures. Studies by Isabel Giraldo and James Traniello, published in the Proceedings of the Royal Society B in 2016, tracked known age cohorts of fidel dentata workers across their approximately 140-day laboratory lifespan. They found no increase in apoptosis, programmed cell death, in the brain. No decline in trail following accuracy. Older workers were, if anything, more precise than young ones. Right up until day 120, these workers nurse larvae with full competence, track pheromone trails without error, and lunge with aggressive precision at intruders placed near the nest. The colony generates no elderly. There are no pensioners. There is no drag on the system from declining individuals. And then, around day 140, something snaps. There is no gradual decline, no warning tremor. The molecular machinery that has held perfect stasis for months undergoes a sudden, catastrophic collapse. The ant dies not from accumulated damage, but from what appears to be a biological timer, silent, precise, and merciless, finally reaching zero. What triggers the switch is still unknown. The gene pathways that maintain this uncanny neurological stability and the signal that finally dissolves it remain an open question in entomological science. She lived her entire life at peak performance, and then she vanished from it. What does it mean to have a perfect mind until the moment you cease to exist? In the ant world, this is not tragedy. It is the design. Level 2, the colony that learned to fear a ghost. A dead enemy held for 5 minutes against the nest wall was enough to change the colony's behavior for 3 days. There are wars in the ant world fought not with soldiers, but with memory. Temnothorax longispinosus is a small acorn-dwelling ant of North American forests. Under normal circumstances, it maintains orderly, territorial behavior, a small colony tucked into a hollow twig or a gap in bark, going about the quiet work of survival. But Temnothorax lives under siege. Its colonies are perpetually targeted by Protomognathus americanus, a slave-maker ant that raids neighboring nests, steals pupae, and forces the hatched workers to labor in a foreign colony, never knowing, chemically, that they are enslaved. Research by Tobias Paminger, Suzanne Foytzik, and colleagues at Ludwig Maximilian University of Munich, published in Behavioral Ecology, revealed something remarkable. A 5-minute encounter with a dead slave maker, not a live one, not a raid, just a corpse pressed near the nest entrance, was sufficient to prime the colony into a state of heightened, non-specific aggression toward all non-nestmates. This primed state persisted for up to 3 days. The colony had remembered. Not individually, not consciously, but chemically and collectively, the ghost of the parasite was written into their behavioral state, translated into a standing order: trust no one. But there is a darker chapter to this story. In colonies that had already been raided, whose pupae had been stolen and raised among slave makers, some workers emerged in a state of profound behavioral disorientation. These rogue workers, host ants raised inside the enemy colony, chemically imprinted on slave maker pheromones, would return during subsequent raids and turn against their own species. They stung and attacked the very workers defending the nest. Their aggression caused more direct harm to their genetic kin than the slave makers themselves. This is not treachery in any human sense. It is what happens when the identity system, built on chemical fidelity, is corrupted at birth. The rogue worker has no betrayal in her heart. She has only the chemistry she was given, and the chemistry was written by the enemy. Level 3. The Shrinking Queen. She wins the tournament, seizes the crown, and then her own brain begins to disappear. When the queen of a Harpagnathos saltator colony, the Indian jumping ant, dies, she does not simply leave a vacancy. She leaves a chemical silence. For as long as the queen lives, her pheromones suppress reproduction in every worker in the colony. The moment she is gone, that chemical leash snaps, and what begins is one of the most radical neurological transformations ever documented in an animal. Workers enter into prolonged, ritualized dominance tournaments, antennal fencing bouts, slow and deliberate, each one a negotiation of status. The winners, those who accumulate enough wins to establish dominance, undergo a total physiological reinvention. Their dopamine levels surge 2 to 3 times above baseline. This hormonal flood initiates a cascade of epigenetic changes. Genes for reproduction are activated, genes for foraging and immune function are suppressed, and then, counterintuitively, the brain shrinks. Research published in the Journal of Experimental Biology by Clint Pennick, Jürgen Liebig, and colleagues, and further developed by Gospodzic et al. In 2021, documented that dominant gamergates, reproductive pseudo-queens, experience a 19 to 25% reduction in brain volume. The mushroom bodies, those seats of learning and memory, contract. The brain conserves metabolic energy for the demands of egg production. The ovaries expand to fill the abdominal cavity. Venom production falls. Foraging ceases entirely. Behaviorally, the transformation is startling. A gamergate, who was once a forager, aggressive, exploratory, responsive to intruders, becomes profoundly timid. She retreats into the nest's interior. She avoids confrontation. She is surrounded by workers who guard and groom her. She has traded nearly a quarter of her cognitive capacity for longevity and fertility. Her lifespan, which would have been 6 months as a worker, now extends to several years. This is not degeneration. It is a calculated biological exchange. But it is also, neurologically speaking, a voluntary diminishment. And it is reversible. If she is policed by nestmates and removed from dominance, the dopamine crashes. The brain begins to grow back. In the ant world, the self is not fixed. It is a chemical negotiation, renegotiated at every moment. Level 4. The ant who never learned to be an ant. Without the colony, the young worker's brain never fully woke up. In the eusocial world, isolation is not simply loneliness. It is a developmental wound. When a Camponotus floridanus worker ecloses, emerges from her pupal case, her brain is not finished. The mushroom bodies, the paired lobes responsible for learning, memory, and the integration of sensory experience, require social stimulation to complete their growth. The tactile signals of nestmates, the chemical vocabulary of the colony, the simple act of being surrounded by others of her kind, these are not comforts. They are neurological inputs without which the brain cannot properly wire itself. Studies by Mark Seed and James Traniello, examining Camponotus floridinus, and parallel work by Boulay and colleagues on Camponotus fella, established that socially isolated, newly eclosed ants show dramatically stunted mushroom body development. Total brain volume remains unchanged. The tissue is there, but the ratio of mushroom body to overall brain volume fails to expand as it would in a socially reared ant. The behavioral consequences are severe and immediate. Isolated ants placed in a novel environment huddle in a small patch and remain there, making little effort to explore. They are, in behavioral assays, more than 10 times less aggressive than their socially reared counterparts. When presented with the crushed body of a nestmate, a stimulus that typically triggers immediate, intense investigation, isolated ants actively avoid the signal, while grouped ants move toward it with urgency. When isolated ants are eventually reintroduced to a colony, They engage in prolonged, almost frantic episodes of trophallaxis and allogrooming, exchanging food and chemicals with nestmates in a rush, as though trying to catch up on a conversation they were excluded from. But the mushroom bodies do not simply bounce back. The window for normal development may have already closed. The ant was never broken. She was simply never given what she needed to become whole. In a species built for togetherness, solitude does not merely sadden, it restructures the brain. Level 5. Born blind in a world written in scent. The gene was removed. One gene, and the world went dark. The ant's world is not visual. It is olfactory. The chemical landscape they navigate, trails of pheromone, the unique scent signature of every nestmate, the alarm compounds of a threatened colony, is as rich and layered as any visual panorama. It is the language in which the colony speaks to itself. At the center of this language is a single molecular gatekeeper, the odorant receptor co-receptor, encoded by the gene orco. Every odorant receptor in the ant's antenna, and there are hundreds, requires orco as its obligate partner to function. Without orco, the antenna remains physically intact, but chemically silent. In 2017, Waring Tribble, Daniel Cronauer, and colleagues at Rockefeller University published a landmark study in Cell using CRISPR-Cas9 to knock out the orco gene in Oacerea bayroi, the clonal raider ant. The results were more catastrophic than expected. Unlike Drosophila fruit flies, where the antennal lobe remains structurally intact in orco mutants, the ant brain collapsed. Approximately 82% of the antennal lobe's roughly 500 glomeruli, the discrete processing units for each odor class, simply failed to develop. The antennal lobe shrank to a third of its normal volume. The olfactory sensory neurons in the antennae degenerated. The revelation was structural, not just functional. The complexity of the ant brain's olfactory architecture is not pre-built. It requires sensory activity to wire itself. Without orco, without the continuous flow of odor information through the developing nervous system, the architecture never forms. Mutant ants were functionally exiled from their own species. They wandered away from the colony. They failed to follow pheromone trails. They refused to participate in collective foraging. They could not recognize nestmates. They achieved zero reproductive fitness. In a world written in scent, they were illiterate. And the library had burned. Parallel findings in Pogonomyrmex barbatus revealed a second layer of genetic disruption. The gene PB4, a CGMP-dependent protein kinase, governs the age-related transition between behavioral roles, from nurse to forager. When this gene is dysregulated, ants may fail to make the transition, becoming trapped in one behavioral mode, unable to shift as the colony's needs demand. To be born without the ability to speak the colony's language is not just isolation. It is a form of neurological exile from a world that will never know you were there. Level 6. When the lock is broken and the enemy walks in, the default state of an ant is acceptance. Aggression must be earned. And when the mechanism that earns it fails, the gates open. The ant colony is a fortress, but its walls are not made of stone. They are made of chemistry. Every ant carries on the surface of her cuticle a complex cocktail of long-chain hydrocarbons, a chemical passport unique to her colony. When an ant encounters another individual, her antennal receptors decode this passport in fractions of a second. If the signal matches, acceptance. If it does not, aggression. The system is a biological lock and key, precise, rapid, almost infallible. But what happens when the lock is disabled? Research from the Zwiebel Laboratory at Vanderbilt University, published in the Journal of Experimental Biology in 2020, demonstrated that pharmacological blockade of odorant receptors in Camponotus species disrupts this recognition system entirely. Ants whose receptors are blocked or overstimulated with synthetic agents lose the ability to distinguish nestmate from intruder. In dueling arenas, they fail to lunge, bite, or initiate any defensive behavior. They default to passive acceptance of foreign ants, competitors who, in a healthy colony, would be met immediately with lethal aggression. The default state, researchers found, is not vigilance. It is openness. Aggression is an active, computed response. When the computation fails, the wall dissolves. The consequences for a colony are not abstract. Without nestmate recognition, social parasites enter freely. Thieves steal brood and provisions. Foreign queens may be accepted and installed. The colony's genetic integrity, its entire reproductive future, becomes accessible to any organism sophisticated enough to circumvent or jam the chemical signal. This finding carries a darker implication for wild populations. Environmental pollutants, synthetic pesticides, herbicides, industrial compounds may mimic these pharmacological blockers. Colonies in contaminated environments may be suffering precisely this kind of chemosensory failure without any visible dramatic symptom. They simply become porous, slowly invaded, slowly dissolved. A fortress without a lock is not a fortress. It is an open door. Level 7. The wanderer with something growing in its head. She was not sick when she left the nest, but something inside her had already decided her fate. In the grasslands and forests of the American South, the fire ant Solenopsis invicta faces an enemy that strikes faster than the eye can follow. The female Pseudactyon phorid fly, a parasitoid of extraordinary precision, locates a worker ant and, in under 1 second, inserts a single egg into the ant's thorax. The ant registers nothing unusual. She returns to her work. The colony continues its business, but the egg hatches, and the larva begins to move. Over the course of days, the maggot migrates upward, working its way through connective tissue into the head capsule. There, it feeds on hemolymph, the ant's blood, and then on the mandibular muscles. Eventually, it reaches the brain itself and begins to consume it. The ant, now neurologically compromised, abandons coherent behavior and begins to wander, aimless, dissociated, moving in loose, purposeless patterns far from the colony. Before the larva pupates, it releases a chemical that degrades the intercuticular membranes binding the head to the body. The head detaches. It falls. Inside the empty head capsule, sealed and protected, the pupa completes its development. Eventually, a new fly emerges from what was once an ant's skull. Research by Rob Plows, Henry Fatamiro, and extensive USTA Ars documentation on Pseudactyon tricuspes and Pseudactyon curvatus confirm the devastating secondary effect. Even ants that have not been parasitized change their behavior in the presence of hovering phorid flies. Hundreds of workers retreat into the mound. Foraging halts. Scouts freeze in defensive postures. Native competitors, unafraid of the tiny flies, move freely into vacated foraging territories. One fly, barely visible to the naked eye, can paralyze the foraging behavior of an entire colony through the sheer psychological weight of its presence. The body is devoured from the inside, and the colony flinches from the shadow of the executioner even before she strikes. Level 8. The puppet that knows it is climbing. Every morning she climbs. Every afternoon she comes back down. She is not lost. She is controlled. Of all the parasitic manipulations documented in the ant world, none is more surgically precise, more climatically calibrated, or more philosophically unsettling than infection by the lancet liver fluke Dicracoelium dendriticum. The fluke's life cycle requires 3 hosts: a mammal, typically a cow or sheep, a land snail, and in the intermediate stage, an ant. When an ant of the Formica genus ingests infective cercariae shed by a snail, hundreds of larvae encyst in the ant's abdomen, sealed in thick-walled cysts, waiting. But one larva, a single individual, does not encyst. It migrates to the anterior ventral region of the subesophageal ganglion, the brain's control center for the mouthparts and the neck. This brainworm does not eat the brain. It programs it. Research by Brian Lund Fredensborg, Simone Nordstrand-Gask, and collaborators, published in Behavioral Ecology, used transcriptomic analysis to reveal that the brainworm dysregulates 1,349 host genes, including core circadian clock genes, genes involved in dopamine synthesis, genes controlling vision, and genes governing odorant processing. The manipulation is not brute force. It is molecular whisper work, and it responds to temperature. When the ambient temperature drops to around 10 degrees Celsius in the cool of early morning or evening, the manipulation activates. The ant is compelled to climb vegetation and clamp its mandibles onto a petal or leaf vein in a rigid, locked grip. She is perfectly positioned to be swallowed by a grazing mammal. As the temperature rises toward 25 degrees, the manipulation releases. The ant descends. She returns to normal nest duties, foraging, interacting, appearing, for all the world, perfectly healthy. She is not dead. She is not in pain. She is simply a vehicle, deployed on a schedule calibrated to the thermal rhythm of the day. Every morning, the climb. Every afternoon, the return. Until the morning she does not come back, the brain worm that controls her will die inside her brain when the definitive host, the mammal, is finally infected. It sacrifices itself. The clonal larvae in the abdomen carry on. She climbs every morning toward a fate she cannot comprehend, controlled by a creature smaller than a cell cluster, executing a plan written in her own genes. Level 9. The living statue that cannot let go. The fungus does not destroy the brain. It does something far worse. It leaves the mind intact inside a body it can no longer control. In the humid forests of Thailand, Brazil, and South Carolina, there exists a fungus with no brain, no nervous system, no centralized intelligence, and yet it executes one of the most precise behavioral manipulations ever recorded in nature. Ophiocordyceps unilateralis, the zombie ant fungus, infects carpenter ants of the tribe Campanotini. And for decades, scientists assumed it worked the way most parasites do, by invading the brain. They were wrong. A landmark 2023 study from Penn State University, published in Nature Communications, used three-dimensional imaging at cellular resolution to map every fungal cell inside an infected ant. The finding was staggering. The fungus does not penetrate the brain at all. Instead, fungal cells spread throughout the hemicole, the body cavity, wrapping around muscle fibers in the legs and mandibles, forming a highly interconnected cellular network linked by anastomosing tubules. The brain floats, structurally untouched, in a body that no longer answers to it. The fungus speaks to the muscles directly. It secretes neuroactive compounds, sphingosine, guanidinobutyric acid, hypoxanthine, near the peripheral nervous system. It depletes leucine in the mandibular muscles, preventing protein synthesis and causing atrophy. It reduces mitochondrial density, starving the jaw muscles of ATP and calcium. The result is the death grip. The mandibles lock, not through neural command, but through physical incapacity. The muscle has been dismantled at the molecular level, frozen in a clenched position. The ant cannot release her jaw because there is no longer a mechanism to do so. But the behavior leading to this moment is equally controlled. Prior to the final grip, infected ants experience periodic convulsions that cause them to fall from the canopy. They leave established trails. They ignore nestmates. Under the influence of what researchers describe as a fungal biological clock, they navigate to a very specific microenvironment, typically 25 centimeters above the ground, on the underside of a leaf or twig, oriented in a specific compass direction, conditions optimal for fungal spore dispersal. Work by Teresa de Becker and colleagues, published in BMC Genomics in 2014, confirmed that the fungus suppresses the ant's immune response genes and alters the expression of host genes linked to the nervous system and circadian timing. After death, the fungus erupts. A sexual fruiting body grows from the base of the ant's head, releasing spores that drift down onto the trails below, where healthy foragers walk, and the cycle begins again. The ant's brain was never touched. She watched, from inside, as something else moved her body to its chosen location, closed her jaw, and held it shut forever. This is not possession. It is occupation, and the occupied never knew the occupier was already inside. Level 10. The spiral and the super colony. Some ants march until they die. Others forget who they are entirely and build an empire without a center, until the day two empires collide. We have arrived at the final level. And here, the disorder is not in a single ant. It is in the system itself. The spiral Eciton burkeli, the army ant of the neotropical rainforest, is blind. Every worker, every soldier, blind. Their world exists entirely in pheromone. They are a collective intelligence without individual awareness, a superorganism that thinks in chemical gradients. Their foraging columns, sometimes hundreds of meters long, function with extraordinary efficiency precisely because every ant does exactly one thing: follow the trail. This is their genius. It is also their catastrophe. When a subgroup of foragers becomes separated from the main column by an obstacle, a fallen branch, a sudden terrain shift, the lead ants may turn back and, in their disorientation, cross their own pheromone trail. A loop is created. The ants follow it. In following it, they reinforce it, continuously depositing home trail and food trail pheromone, amplifying the signal. More ants join. The loop tightens, and soon, hundreds or thousands of army ants are marching in a perfect circle, with no exit and no salvation. The naturalist T.C. Schneerle documented a case in 1944 where a circular ant mill measured over 370 meters in circumference. The ants march for hours. They march for days. They cannot break the feedback loop, because the feedback loop is stronger than any individual's hesitation. They march until they fall from exhaustion, dehydration, and starvation. The entire foraging column destroys itself. It is not madness. It is mathematics. A self-organizing system. optimized for following, that encounters a condition it was never designed to escape. The supercolony on the other side of the world, in the hedgerows and coastal meadows of Mediterranean Europe, a different kind of collapse is unfolding. Slower, quieter, and in some ways, more philosophically troubling. Linapithema humul, the Argentine ant, is one of the most successful invasive species on Earth. In its native South America, Argentine ant colonies behave as colonies should: distinct, territorial, aggressive toward neighboring nests. Their chemical identity, encoded in cuticular hydrocarbons produced by genetically diverse populations, varies enough between colonies that nestmate recognition functions reliably. Workers know who is family and who is not. Then, in the 18th and 19th centuries, small numbers of Argentine ants began to travel with human commerce. They arrived in Europe, North America, Australia, Japan. And something happened. The founder populations were tiny. The genetic diversity they carried was minimal. At the locus governing cuticular hydrocarbon biosynthesis, the very genes that produce the chemical passports of nestmate recognition, the variation was nearly erased. Colony after colony, in the introduced range, produced the same chemical signature, the same smell, and so the walls came down. Workers from nests hundreds of kilometers apart, meeting for the first time, did not fight. They had no reason to. They smelled the same. A single super colony emerged along 6,000 kilometers of the European Mediterranean coast. A unified, borderless, continuously interconnected population of billions of ants with no internal aggression, no territorial limits, and no mechanism for distinguishing kin from stranger. The genetic relatedness between distant super colony members is effectively zero. Kin selection, the mathematical logic that underpins social cooperation, no longer applies. Research by Nathan Tsutsui, Thibaut Giraud, and colleagues, published in the Proceedings of the National Academy of Sciences and peer-reviewed journals from the year 2000 to 2003, confirmed the scale and the mechanism. Evolutionary models by Matesser and colleagues in 2024 have explored the destabilizing long-term dynamics. The supercolony is not a utopia. It is a recognition system that has failed at catastrophic scale. Resources are monopolized. Native ant species are obliterated. Ecosystems shift. And when two distinct supercolonies meet, genetically cleansed lineages that descended from different founder events, and therefore carry slightly different chemical profiles, the contact zone becomes a battlefield. The ants fight with near 100% mortality in dueling zones. The colony, so unified inside, becomes a war machine at its edges, expending enormous energy in relentless, unresolvable border conflicts. The spiral kills with feedback. The super colony kills with identity. Both represent the same fundamental truth: a system optimized for one condition will, under different conditions, devour itself. The ant that cannot stop marching and the ant that cannot remember who she is, they are mirror images of the same vulnerability. Precision, taken to its limit, becomes its own undoing. 10 levels, 10 conditions. From the worker who lived perfectly until the moment she didn't, to the colonies that lost themselves so completely they became something evolution never intended. What does the ant know of any of this? Nothing. She knows the trail, or she doesn't. She knows the nestmate, or she doesn't. She follows the pheromone, or she circles until she falls. Her experience, if she has one, is wordless, immediate, and total. There is no narrative she can tell herself about what is happening to her brain. In that silence, science steps in. And what it finds, again and again, is that the line between function and dysfunction in living systems is not a wall. It is a threshold, a chemical gradient, a degree of temperature, a single missing gene, a loop in the trail. We study the ant not because she is simple, but because she is not. We study her because in her tiny, intricate, perpetually fragile brain, we see the same truth that governs all nervous systems. Complexity is always one disruption away from disorder. The colony marches on. For now, the mind, any mind, is not a fortress. It is a negotiation, held together by chemistry and circumstance, and renewed with every heartbeat.